everyone. Today we are going to talk about gene therapy and cell therapy. First, let us know the basics. A gene is the basic physical and functional unit of heredity. Genes are commonly made up of DNA. Some genes act as instructions to make molecules called proteins. Humans have an estimated number of genes between 20,000 and 25,000. How does gene therapy work? Gene therapy is designed to introduce genetic materials into cells to compensate for abnormal genes or to make a beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. Cell therapy, on the other hand, is the introduction of new cells into a patient's body to grow, replace, or repair damaged tissue in order to treat a disease. What are the uses of gene therapy? Gene therapy has many uses, which includes replacing missing or defective genes, delivering genes that speed up the destruction of cancer cells, supplying genes that cause cancer cells to revert back to normal cells, deliver bacterial or viral genes as a form of vaccination, providing genes that promote or impede the growth of new tissues, and delivering genes that stimulate the healing of damaged tissues. What are the common types of cell therapy? The most common type of cell therapy is blood transfusion and the transfusion of red blood cells, white blood cells, and platelets from a donor. Another common cell therapy is the transplantation of hematopoietic stem cells to create bone marrow, which has been performed for over 40 years. What are the strategies being used in gene therapy? First, we have gene editing. Instead of fixing words, gene editing rewrites DNA, the biological code that makes up the instruction manuals of living organisms. Next, we have cell programming. Cell programming is the process of reverting mature, specialized cells into induced pluripotent stem cells. Cell elimination. When genetically different cell populations are mixed, there is a competition between cells such that losing cells are eliminated from a tissue. Such cell elimination is also observed during normal development in genetically homogeneous tissues. What is the role of viruses in genetic therapy? Viruses are used as gene delivery vectors. Viruses can be modified in the laboratory to provide vectors that carry corrected therapeutic DNA into cells where it can be integrated into the genome to alter abnormal gene expression and correct genetic diseases. This involves removing the viral DNA present in the virus and replacing it with the therapeutic genes. Can viral vectors make you sick? The answer is no, because these viruses used in gene therapy are designed to reduce the risk of adverse effects. These viral vectors are tested in animals and in human trials to make sure that human cells are not harmed. The conclusions from these trials imply that gene therapy has the potential to cure numerous genetic diseases and that the procedures appear to have minimal risks to the patient. But the efficiency of gene transfer and the expression of the corrective genes in the human patients is, as of now, still very low.